Okay. So good evening. My name is Dr. Shakti, and we're going to be continuing with our revision program that is happening right now. We're going to leaf through the different topics that have been covered in the past few months, and we are going to look at it in a new perspective where we're going to rely on the important questions, most often asked questions that keep coming up. And we're going to delve more into that and help you understand how exactly you're supposed to write these answers. Okay. So you can just take a look at some of these questions over here. So this is essentially the core of what we'll be discussing today. You can see here one long essay. This is very often asked as a question. Analyze the transport mechanism across the cell membrane. And they're in fact mentioning how to do this by discussing on diffusion, osmosis, active transport, facilitated transport. And then they're asking you to highlight the physiological significance of each type of in terms of your nutrient and waste management. So the thing is, a question like this in your first semester exam, it will not be asked, because to answer this last part, you need to have studied your GIT, parts of your renal system, entirety of your renal system, in fact, in order to be able to more effectively write that answer. So for your first semester, no, a question like this will not be expected out of you mostly. Okay. But for your final exam, it can be asked, in which case you will have to link different chapters, things that you learn in your GIT, renal system, anywhere else, and try to bring it together. Okay. Now we can see here short essays also. This is a very, 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 very important question that keeps coming up from your general physiology. That is sodium potassium ATPase. What is sodium potassium ATPase? How does it function? And there are certain things that you need to always mention when a short note on sodium potassium ATPase is coming. We'll discuss that. And then you can see here ion channels in nerve and muscle. So this is actually something we're going to be discussing in the next class when we're talking about nerve muscle physiology, but certain concepts about your normal diffusion, that is the basis of this. Then we have GLUT transporters, which again, once you do your endocrine system, you'll know it better, but I just mentioned it today also, in case a question like this is asked. Then short answers. What is again electrochemical gradient, and why is it crucial for neurotransmission? Define the term exocytosis. So apart from these transport mechanisms we saw, like diffusion, osmosis, active and facilitated diffusion and all, you have something else like exocytosis or endocytosis. These are again some other questions that can come separately. And explain secondary active transport. How is it different from primary active? So difference between is your primary active transport and a secondary active transport is one question that often comes. Difference between a simple diffusion, facilitated diffusion is another question that is often repeated. So to sum it up, a long question of the entirety of transport mechanism can come. Sodium potassium ATPase can be specifically asked as an important short note. Then we have difference between primary active and secondary active transport, difference between your simple diffusion, facilitated diffusion. And then each of these can be separately asked as a short note. Write a short note on diffusion. Write a short note on active transport. Then we talked about exocytosis, endocytosis. So these are all very important questions that keep coming from this topic. And if I had to choose one basic thing that you need to study, that is going to be a sodium potassium ATPase. Let's go on to understand how to write these answers. So I usually show these kind of pre-made notes for revision classes because I want you to understand how to write points for the exam along with diagrams and how to give a basic stratification. Because if that stratification is there, then you get marks. Otherwise, if you just write like pages and pages, and it's going to be difficult for the examiner. So I'd always want you guys to write in a point form, bullet points as to what the organization is, so that the examiner knows that, okay, you have everything covered and can give you marks adequately. So first thing, diffusion. What is diffusion? It is a passive movement. So basically, when concentration of substance is higher on one side, and there is some sort of a semi-permeable membrane, then that is because of the Brownian movement, higher kinetic energy on one side, that will result in the transport of substances across from a high concentration to a low concentration without the use of any energy. That is your diffusion. It is a passive process. Now, diffusion can be three types. We have your simple diffusion, we have your facilitated diffusion, and we have non-ionic diffusion. So if a short note on diffusion comes, you'll have to talk about 1, simple diffusion, 2, facilitated diffusion, 3, non-ionic diffusion. Before that, define what diffusion is. So define diffusion, then simple diffusion, facilitated diffusion, non-ionic diffusion. And we're not done yet. Under this short note, usually you have to talk about Fick's law. You write these 5 things, then that is how you get full marks for that short note. So we'll delve on to what is simple diffusion. Under simple diffusion, you can talk about how across the membrane, your lipid-soluble substances can directly pass through from a higher concentration to a low concentration. The lipid-soluble substances, for example, these gases and steroids. Now, the other substances will usually require, ions especially, will require some sort of channel or pore. So usually the pores are ungated, and once it is active, it just keeps going in one direction. Then we have gated channels, which are particular ion channels. And this sort of gating requires different mechanisms to activate it. What are the gating mechanisms? Either a ligand from outside can come and bind to it. Changes in membrane voltage can open it up. Some form of phosphorylation can open it up. It can even be a mechanical gated or some chemical or enzyme. So these are different ways by which gating can happen. And such gated ion channels can be there. Ungated pores can be there, like porin, perforin, echoporin, all of these can be there. Or through the lipid membrane directly, if it is lipid soluble. These are all examples of simple diffusion. So what did you understand here? First, you have to talk about one particular thing, simple. You give a diagram for it if possible. And under that, you give what are the different types and give examples of each of this. So this is how this answer needs to be written. And then we have facilitated diffusion. In facilitated diffusion, differences, there is going to be a carrier protein. Not like a channel or pore that we see here. Instead, it is a carrier protein. What does it do? It's not like something that, which, as you can see over here, it is just open like this. And there is free flow of ions. It's not something like that. Instead, what is it? It is a carrier protein, which undergoes conformational changes. How does it undergo conformational changes? In one state, can you see over here how it is open on one direction, that whatever substance is coming in, binding to a site, and then resulting in a second configurational change in between state. And then what is going to happen? From here, this is going to shift to the third state, resulting in movement of this substance outside. So this is something that happens with the help of a carrier protein. So facilitated diffusion uses a carrier protein like this. What are the examples? So you draw a diagram, explain how facilitated diffusion happens, and then you give examples. One common example is your blue transporters. glucose, fructose are all transported using GLUT transporters. Then you have urea transporters, certain organic cation transporters. So GLUT transporters, urea transporters, organic cation transporters are all examples of your facilitated diffusion. Now remember that question I was telling you earlier. What is the difference between simple diffusion and facilitated diffusion? So under that you have to, if that is the question that is asked, then you have to talk about how in simple diffusion, down the concentration gradient, either to lipid soluble or through pore gate, and give examples. Here also mention how there's a carrier protein involved and give examples. But after that, I want you to draw this particular diagram. This is the rate of flow. A rate of flow for the concentration. When the concentration of that substances increases, the rate of flow generally increases. As more concentration gradient is there. For example, this is the number of substances on this side. This is the number of the substance on this side. Instead of that, if the number of substances keeps increasing, the rate of flow keeps increasing. That is exactly what is shown here. As the concentration increases, the rate at which it's flowing is increasing. This is your simple diffusion. In facilitated diffusion, however, you see that this curve at some point flattens. It is reaching a maximum rate. Beyond this maximum rate, it cannot go. That is because the carrier proteins have been saturated. There is no more available carrier protein in order to carry that substance. It reached its maximum where at that maximum speed, all of the carrier proteins are utilized. So this one difference here, rate of flow and concentration gradient. That is something you need to draw and explain. So you explain what is simple diffusion, what is facilitated diffusion, then give examples, then third, explain about the saturation kinetics, and if possible, draw the diagram. Then you will get good marks if this question is asked. Now the third type of diffusion is non-ionic diffusion. So in non-ionic diffusion, what happens? Your H plus and HCO3 minus usually cannot cross the membrane because it is ionic in nature. And if it does not have its particular ion channels, it cannot cross inside. But what does it do? Instead, it goes to H2CO3, becomes H2CO2. CO2 is now lipid soluble and can go inside. And once inside, it can ultimately split into H plus. So it's as if that H plus, HCO3 minus has gone inside by converting to another form. So this is what is called a non-ionic type of diffusion. So this kind of diffusion example is this H2CO3 using that, or your ammonia through NH4 plus, even short chain fatty acids for that matter in your intestine absorption when it's taking place. So these are all examples of non-ionic diffusion. So if a question of diffusion is coming, you have to mention all three. Okay. If a difference between simple and facilitated, you know what to write now. And if a short note on diffusion is coming, at the end of it, talk about your Fick's law. What is Fick's law? That rate we were talking about. So if this is the concentration of a substance over here. Okay, this is the amount of substance on one side. This is the amount of substance. As the number of substances increase on the one side, the concentration gradient increases, more will be the rate of flow. This just now we are talking about. So it's directly proportional to change in the concentration gradient. What about the area over here? Greater the area through which it is diffusing, greater the rate of flow. So that is also directly proportional. On the other hand, if the membrane thickness becomes very high, then what happens? The thickness of this membrane through which it is passing. Okay, it's passing through a membrane, and that membrane is more thick. Then it becomes more difficult. So that is inversely proportional. So Fick's law talks about your net flux, that is proportional to delta C A by T. Okay, that is your Fick's law. So with that, we cover one question, that is your diffusion. Next is your active transport. Active transport are of two types. We have primary active transport, secondary active transport. What is different about active transport? Movement of ions from a low concentration to a higher concentration. Again, the concentration gradient has to go. For that, it has to utilize ATP. That is active transport one. That is divided into two types: primary active transport, secondary active transport. What does primary active transport mean? Whatever mechanism is involved here, that by itself utilizes ATP. So there is one transporter here. Okay, there is one channel here. That channel by itself utilizes ATP. And then that by itself takes things against the concentration gradient. That is the primary active transport. Secondary active transport, what does it do? This does not by itself utilize ATP. There is something else over here that utilizes ATP. And whatever changes happen as a result of that, because of that, it will now take certain other substances across the concentration gradient. That is example of a secondary active transport. Okay, so short note on active transport is coming. You have to talk about what is active transport. Then under that, talk about primary active transport, then secondary active transport. If the differences between primary active and secondary active is coming, you start out by describing these two differences. This difference that I just mentioned. Then give examples. So primary active, usually we talk about P type, F type, V type. P type, your sodium potassium ATPase. This is again a very important question. So sometimes again, primary active transport by itself can come as a short note. Okay. Sodium potassium by ATPase by itself can come as a short note. Now, P type primary active transporters. One example is sodium potassium ATPase. Then we have hydrogen potassium ATPase, which is there in your GIT, proton pump for acid secretion. Then in your muscles, we have something called SERPA, sarcoplasmic endoplasmic reticulum calcium ATPase. What does it do? Calcium is actively taken back inside the endoplasmic reticulum. That is your P type. Then we have F type, which you learned in biochemistry, the FOF1 particle. So in chemiosmotic model, yesterday we were talking about it. We have the F type, FOF1 particle, through which H plus comes in, and because of that, ATP is generated. And then we have V type, that is vesicle type, especially in certain vesicular formats where acidification is required. A hydrogen ATPase is present. This you especially see in certain cells like lysosomes and all that. V type ATPase can be there. Okay, or even osteoclast mechanism. In that also you see these V type ATPase. So generally these are where some sort of acidification is required, where you actively take in H plus. So these are examples of P type, F type, and V type. This specifically we have to talk about P type. Okay, so if sodium potassium ATPase is asked as a short note, what are the headings under which you have to cover this? One, talk about its structure. What is the structure of sodium potassium ATPase? Two, how is the mechanism of that? What are the functions of sodium potassium ATPase? What are the factors regulating? You can even get a long question of transport mechanism, and it can be subdivided into sub-questions. Some of them can ask you, what is the mechanism of sodium potassium ATPase? 2 marks. Functions of sodium potassium ATPase, 2 marks. Factors describing sodium potassium ATP, affecting sodium potassium ATPase, another 1 mark. And with a neat label diagram, draw sodium potassium ATPase. That is another 1.5 marks. Okay, another half mark for some clinical application. So we have a long question there. Okay, that's how sodium potassium ATPase is very important. And these are various heading you have to cover it under. Again, structure, what is the mechanism, what is the function, what are the factors regulating. So in structure, you have to draw two subunits, an alpha subunit, a beta subunit like this. Okay, we draw alpha subunit, we draw beta subunit. This alpha subunit has two areas, where one is where sodium is binding, the other is where potassium is binding. Where potassium is binding, next to it, draw a small area called obeying binding domain. It's a drug that binds to this area. Intracellularly, there is an area where ATP is converted to ADP and inorganic phosphate. So that is the area where your ATPase activity is there, over here. Okay. Then this inorganic phosphate will come and bind to one particular point. And you can draw some carbohydrate residues on top of your beta domain. So this is your structure of your sodium potassium ATPase. Now, how does sodium potassium ATPase act E1, E2 format? So it has two states in which it is existing. One is E1, the other one is E2. So initially it is there in the E1 phase where it is open downside over here. So what is going to happen? Sodium is going to come and bind to it. And in the E1 phase, ATP is already attached to it. That is how it becomes an E1 phase. Okay. Now sodium comes. How many sodium? 3 sodium. And at that point, E1 gets changed to E1P, where ATP hydrolysis happens and that inorganic phosphate is the extra. So it's a phosphorylated E1. So that is called E1P. Now this E1P state shifts to an E2P state, where the opening is no longer here. Instead, it becomes on the opposite side. So now sodium is able to move outside. So sodium goes outside in the E2P phase. Now, in the E2P phase, what will happen? The phosphate group will go away. And now, because of that, the E2 phase is with an opening on this side. And it is in this phase that your potassium comes inside. Now, potassium comes in. It is existing in E2 phase. That is when ATP will come and bind to it. Once ATP binds, this E2 phase gets converted to your E1 phase. Back to where it began, and instead the opening is going to be over here. So what is going to happen? The potassium is going to come. The 2 potassium from outside reached inside. Now again, it becomes the E1 phase, because of which sodium will now come in. That ATP gets hydrolyzed, it becomes the E1P. Then E1P becomes E2P, hence resulting in opening on the opposite side and sodium coming out. So this whole cycle repeats. So this is how we can explain the mechanism of how sodium potassium ATPase acts. Next, what are the functions of sodium potassium ATPase? One, it is an electrogenic pump. What does it mean? 3:2 ratio. So you saw how the 3 sodium is going outside, 2 potassium is coming inside. So it's not actually maintaining the ratio. 3 positively charged ion is going outside, 2 positively charged ion is coming inside. So it is maintaining some sort of a negativity state on the inside, because one extra positively charged ion went away. This is very important in order to maintain your resting membrane potential in the negative zones. Okay, so it is electrogenic in nature. It helps maintain the volume. One extra sodium is always given outside. Otherwise, what will happen? The sodium which is there, it keeps accumulating on the inside. And with more sodium inside, more water is going to come in, it'll swell up, expand. So in order to maintain your volume, you can use the sodium potassium ATPase to control the amount of sodium which is there inside. At rest, what do you call the basal metabolic rate? your energy utilization in a cell, 24% of the energy utilization in a cell is through your sodium potassium ATPase. In certain cells, for example, neurons, this energy utilization can go as high as 70%. And lastly, it helps provide a gradient for the secondary active transport. So remember when I was telling you earlier, over here, I was explaining how secondary active transporter requires some other thing to actually utilize the ATP. That is often your sodium potassium ATPase. Most often, the example of that is going to be a sodium potassium ATPase. So, sodium potassium ATPase is acting in order to provide a gradient for secondary active transport. These are all the different functions of your sodium potassium ATPase. Then, what are the factors that affect it? So, factors that increase, factors that decrease. So, thyroid, what does it do? It increases the basal metabolic rate. One of the ways it does that is by increasing the activity of sodium potassium ATPase and the mitochondria. So, thyroid is one thing that increases. Insulin, aldosterone also seems to have a way of increasing your sodium potassium ATPase activity. On the other hand, dopamine digitalis inhibited. If there's hypoxia, hypothermia, and if certain metabolic poisons like 2,4-dinitrophenol, they can inhibit and reduce the activity of sodium potassium ATPase. Now, this is something that can sometimes come as a question. Digitalis. Its function of blocking sodium potassium ATPase. How does it help in your cardiovascular system? So that is where in cardiovascular system, there is a transporter, which is exchanging sodium for calcium. Okay. So what does your sodium potassium ATPase do? It takes sodium outside and brings potassium inside. But in effect, sodium has reduced inside. So this will be a good avenue for sodium to come in exchange for calcium. So this is a sodium calcium exchanger. So if your sodium potassium ATPase is inhibited, what will happen? This gradient is no longer there, which means sodium will not come in, which means calcium will not go out, which means there is higher calcium inside the cell. Higher calcium means more force of contraction. So it helps the heart contract better in heart failure and all that. Okay, that is one clinical application of this. So again, sodium potassium ATPase is a very important question. You talk about the structure, the mechanism, the function, and the factors regulating it. Then we go to secondary active. Secondary active, we talk about it with regard to two categories. We have your symporter or co-transporter, or other words, opposite is antiporter, exchanger, counter-transporter. Symporter, co-transporter, same direction it comes. Opposite direction it moves, antiporter, exchanger, counter-transporter. So we already saw an example of a counter-transporter. We saw how sodium potassium ATPase will act and result in sodium coming in, calcium coming out. We just saw it. That is one example of an antiporter or an exchanger or counter-transporter. So one example is sodium calcium exchanger. Similarly, there can be a sodium hydrogen exchanger. There can be a chloride bicarbonate, which we call an anion exchanger. These are all examples. So with diagram, give examples. Okay, this is example of your antiporter, exchanger, counter-transporter. What about symporter? It goes in the same direction. So sodium is coming in the same direction. And along with that, glucose will also come in. So glucose will come against the concentration gradient. In a similar way, calcium went out against the concentration. That is why it's called secondary active transporter. Okay, please remember that. So glucose coming in, this is an example of sodium glucose co-transporter, which is there in the kidney, SGLT. It is also there in your intestine. Then we have sodium amino acid can come in together. Sodium iodide can come in together. Okay. Apart from that, in your renal system, sodium chloride co-transporters, sodium potassium 2 chloride co-transporters. These are all important examples of co-transporters. Okay. So remember examples, at least few of these examples you remember and explain them. Sometimes a question of differentiate between a co-transporter and counter-transporter can also come. So remember that first important essay, we talked about your diffusion, we talked about your transport. Under that, you talk about simple diffusion, facilitated diffusion, we talk about non-ionic diffusion, we talk about primary active transport, we talk about secondary transport, importer and antiporter. And that covers that huge essay. Next is certain other components, like exocytosis, endocytosis, and all. Okay, before that, I just want to show you this one table from Gano. So these are various transporters. You can see here sodium glucose transporters. So that is an example of a secondary active transport. Sodium glucose transporter is an example of a secondary active transport. So this SGLT2 something that is there in renal tubule. GLUT is an example of a facilitated diffusion, a transport protein, carry protein. Normally in your certain areas like the brain and all, GLUT1 is one way by which glucose absorption is taking place with the help of a transporter. But otherwise, the rest of the body requires skeletal muscle, cardiac muscle, adipose tissue, other tissues require insulin to take in glucose. That is what we call a GLUT4 transporter. That is why the insulin is a pretty important hormone in our body. And then we have another GLUT2, through which your pancreas should detect that, okay, glucose is coming from outside. How does it detect? Through this GLUT2. Only then it releases insulin. So first pancreas has to detect. That is through your GLUT2. Okay, so GLUT2 is one way by which your pancreas detects glucose and takes it in. Rest of the tissues taking it in is GLUT4, and that needs your insulin. GLUT1 is an example of your basal transport. GLUT5 is one way by which fructose can be transported. Okay, so just to give you an idea overview of your GLUT transporter, because one question on GLUT transport was shown over there earlier. Now coming back to the next point that you need to learn in this chapter. Okay, so till now what we covered is the most important part that you have to study before the exam and go. This sometimes it is asked, sometimes it is not asked, not as important as the one. Nonetheless, questions do come from it. So two things, exocytosis, endocytosis. Okay. So again, this is the overview. Endocytosis, you can divide it into different types. One is phagocytosis. If the substance is larger than 0.5 micrometer, we call it phagocytosis. Then we have pinocytosis. Between 0.2 to 0.5 micrometer, it is macropinocytosis. Less than 0.1 micrometer, it is micropinocytosis. And then there are non-phagocytotic pathways, which include some clathrin, caveola mediated mechanisms. Okay, so first we'll go to your exocytosis. Again, one question we had seen over the exocytosis. So in exocytosis, you talk about two different ways of secretory pathway, a constitutive and a regulated mechanism. So the vesicle, which is budding off from your Golgi apparatus, that can either be coated with a substance like clathrin or a coaterma. And it will result in exocytosis. Okay, this difference is not important at your level, I think. Okay, not many questions actually come from here. So I'm just going to go to the next place. This one structure. So the vesicle finally, vesicle has a V snare attached to it. Okay, that is a synaptobrevin, a V snare protein is there, synaptobrevin. And the membrane over here has a T snap. This is usually called syntaxin. Okay, in terms of your transmission across the nerves. Okay, again, that also, remember we had a question over here. Neurotransmission, exocytosis, role in neurotransmitter release. Okay, that is why I'm specifically mentioning. So in neurotransmitter, these V snare is called synaptobrevin, T snare is called syntaxin. And attached to it is another protein, which is RARGTP. This is, you can think of it like a molecular switch, which regulates the activity of the fusion complex. Okay, so usually when calcium comes in, it binds to another substance over here called synaptogmin. That results in a configurational change, resulting in V snare, T snare proteins with RAGTP to combine and form a ternary complex. And then with another protein called synaptophysin, it helps in fusion and release. This is how exocytosis is happening. It starts off with calcium coming in. Calcium comes in, synaptogmin, then V snare, T snare, RGTP forming a ternary complex, resulting in fusion with synaptophysin and release of the contents of vesicles. That is not done. After that, this has to disassemble and go back. For disassembly, you have two other substances, which is NSF and SNAP. So this NSF utilizes ATP. And with the help of NSF and SNAP, it results in deassembly of it and takes it back up. So this is the entirety of how exocytosis is happening. And you mention exactly how it is happening in your neurotransmitter release. With synaptogmin, synaptobrevin, syntaxin, synaptophysin, all that. That answers that question. Next is endocytosis. We already saw the flowchart of what is the different types of endocytosis. Of that, phagocytosis, if specifically a short note is coming, what are you going to do? So phagocytosis, you have to understand the steps of phagocytosis. You already know 0.5 micrometer. Above that. First, there should be some sort of a detection. Okay, so what is going to happen? Your cell will have some sort of a receptor protein which will detect a ligand. That is the first step, detection. Once detection happens, then your internalization happens by pseudopod formation. Once internalization happens, this will be in a small vesicle called phagosome. The thing is, phagosome is in a special way such that not much of the fluid, this entirety of this area is filled with fluid outside, extracellular fluid. So when phagosomes are formed by internalization, there is no fluid in that phagosome. It's only the particle. Very little fluid. Okay, if fluid is involved, we call it pinocytosis. So without fluid as a phagosome. Now phagosome, what happens to the phagosome? Your lysosomes degranulate and it forms what is called a phagolysosome. and it results in destruction of that. So these are the steps in phagocytosis. So with diagram, I want you to explain each of these steps in phagocytosis. What about pinocytosis? Pinocytosis, we have macropinocytosis, micropinocytosis. Again, what are the difference? The difference in the size of the object. 0.2 to 0.5. Less than 0.1 is your micropinocytosis. In macropinocytosis, what will happen? The substance, one branch of actin polymerization happens and internalization happens like that. And it comes in, but it is filled with fluid. And it's a larger vesicle. In micropinocytosis, it's going to be a smaller vesicle. No such pseudopod formation is required. It directly keeps coming in. And there is some degree of clathrin, caveolin, or clathrin caveolin independent mechanisms involved in forming these vesicles. That is micropinocytosis. Okay. Now, lastly, they might sometimes ask you what is clathrin-mediated, caveolin-mediated pathways. So, in clathrin-mediated pathways, you talk about a structure called triskelion. Okay, so what happens? This the substance that you saw over here, it comes and attaches to it. It is on that membrane. This structure that you see over here, a triskelion kind of a structure that keeps coming and attaches to this, forming a lattice network like this. Multiple of them will come and attach to it. So imagine multiple of them keeps attaching into it, and with the help of a dynamic, it eventually goes like this. It's all, completely covered with this lattice network of triskelion structures. And with dynamic, finally, it'll pinch off. It'll cut over here and it'll pinch off into a globe like this. And it is still attached to these proteins, clathrin coated. It uses ATP now to lose this cage of proteins. Okay, and usually it goes into a substance like endosome. There'll be a endosome, and this will go into an endosome, where retrieval of parts of this cage will be taken back outside, and whatever substance was taken inside with its receptor. So the receptor will be here now. That will be different. The substance will be different. Your triskelion structure will be different. Okay, now whatever required, this will be taken back, receptor will be taken back, this also will be taken back, and whatever needed to be done to this will be done. This is how clathrin cage is activated. In a similar way, there is another caveolin-mediated mechanism. There is a lipid-rich area in the membrane. Okay, in that lipid-rich area, usually on top of it, a GPI-anchored protein will be there. And below that, we have your caveolin protein. This again, through this caveolin protein, it will again, in a similar fashion, will keep budding off. Dynamin again will act, different type of dynamin, but dynamin again will act, bud it off. Now, after budding off, instead of going to endosome, this goes into a separate complex called caveosome. This is just a difference between clathrin-mediated mechanism and a caveolin-mediated mechanism. So if questions of endocytosis comes, you have to talk about phagocytosis, you have to talk about pinocytosis, you have to talk about clathrin-mediated endocytosis, caveolin-mediated endocytosis. Okay, if exocytosis is coming as a question, I want you to draw and describe steps in exocytosis. So that deals with a very important topic that we had to cover today. Okay, so let's just go to understand why you need to choose Auricle. Auricle has, the foundation classes which have been pre-recorded and kept for you to read at your own convenience and go through it. We have the live sessions, which will give you, interaction with the examiner, the teachers, and you'll get the feel of actually being in that class. We have the express exam module, which is just the revision of whatever is being taken in the foundation course, but for you to prepare for the exam. Important points, just like how today's class was, the entire syllabus is covered under 20 hours. And we have 1-to-1 doubt clearing sessions, hard copy notes which are made available, and there is also question banks of various difficulties. Even the practical sessions have been pre-recorded and kept in the app. So these are the various subscription plans which are made available. You can use my code Dr. Shakthinag 20 for discount on subscription. So thank you for watching. You can contact Team Auricle if you want through these various social media handles as well. Okay.